Hey everyone, this is Patrick. Welcome to SYNB0. This is Introductory Synthetic Biology, version number 1. This is going to be an online lecture series by CSBERG. CSBERG stands for Canadian Synthetic Biology Education Research Group, and what we do is develop educational material for synthetic biology. So this is the first version, as I said. We're going to be creating better versions in the future, but we want to create this material for the iGEM teams out there in Canada and in the world, and for people who are interested in synthetic biology in general. In this lecture, this is the overview of SYNB0, and we're going to talk about some very introductory stuff to kind of segue into our more specific topics. So in this first lecture, we have 4 sections. In section A, we're going to briefly talk about what CSBERG is, who our instructors are, I'll introduce you guys, and our relationship to iGEM Toronto and Biosynthesis, and how we're inspired in our topics from these groups. In section B, we're going to talk about how to define synthetic biology, or SYNB0, and this is a really important topic just because there are so many ways to describe or define synthetic biology because of how many different disciplines helped fed into its growth to become what it is now. And so we're going to walk through how to define synthetic biology and come up with a working definition for this course. And hopefully you can kind of see how comprehensive and all-encompassing this working definition is. In section C, we talk about the Artemisinin project, where it's related to mosquitoes, farmers, and how biofuturism actually looks like right now. And so the Artemisinin project is a project done by Jay Kiesinger and his lab, where they tried to produce this anti-malarial drug called Artemisinin semi-synthetically using yeasts that have been genetically engineered. And so it's really important to understand and know what this Artemisinin project is, just because whenever people talk about the economic impact or the future investments in synthetic biology, they like to refer to the Artemisinin project as a key example of going from bench scale lab work all the way to industrial production at Sanofi, the pharmaceutical company. And lastly, in section D, we're going to introduce the 3 streams for wet lab, dry lab, and human practices. This is inspired from iGEM. And so we're going to talk about the different topics we will kind of explain throughout these different modules. And yeah, so let's get started. First of all, again, I'm Patrick. I'm a PhD student in Biozone. And so I'm the head instructor of this team of instructors. Now, just because I'm the head instructor doesn't mean I did everything. I really depended on the people and their domain expertise to help me develop this material. And so hopefully you'll look into their material and learn from them too, and not just me. I'm also the program director of CSBERG. We do more than just teaching material, but I want to introduce what CSBERG is to you guys. So the 2 questions that drive me philosophically insane is what is CSBERG and what does CSBERG actually do? It's a philosophical question for me just because the way I started CSBERG was based off of a philosophical question, which was, there is this insufficient, this lack of pedagogical research in SynB0. And so what can we learn from traditional pedagogical research that has already been done for STEM fields? How do we bring that innovation and that understanding, that insight to the field of synthetic biology so that we can teach synthetic biology better, we can have better student experiences, we can have more useful content being delivered to students that prepares them for academia, for industry. How do we teach this thing better? And so what we do in general is we're trying to lay the groundwork within Canada to have better synthetic biology education to train the future synthetic biologists according to what is needed in academia and what is needed in industry. And so more specifically, this is what we put on our website, what CSBERG is. We're a research group comprised of post-undergraduate and graduate students, and we're trying to develop best practices for teaching synthetic biology. That is kind of our main goal right now. And so a lot of the work we've done already, it has been related and developed from work we've been giving and putting into the iGEM Toronto team to train them, to give them educational material. And so you can learn more about what CSBERG is at our website through that URL over there. And more specifically, what do we do? Well, I mentioned before that CSBERG is kind of a response to the lack of pedagogical research. So what we're doing is kind of 3 parts right now. In the first part, what we want to do is understand the status of the existence of iGEM teams in Canada. And so we surveyed electronically and through interviews the iGEM teams across Canada. And so we're doing a study from this. We're preparing a manuscript to kind of describe what we see in the iGEM landscape of 2019 and to make recommendations for how to improve support for iGEM teams and how iGEM teams can improve themselves to be more sustainable and to better teach their members. And point number 2, this is what we're familiar with right now. We're developing curricula, is really what it is, to teach synthetic biology to what we want is to be the senior undergraduate student at that level. So SYNB0 is the online version. We wanted this to be taught physically to U of T students in the form called SYNB1, but we kind of reversed and we said, okay, let's produce online content because of the whole COVID situation, and then we'll teach SYNB1 when it's time, when fall term or winter term opens up. And point number 3 is more specific for the University of Toronto. We want to develop the SynB0 community here, the wet lab, dry lab, and the human practices people. We want to develop all of it, and not just specifically experimental people. And so we have SYNB2, which is our initiative to kind of get the ball rolling on developing this network. So I want to introduce CSBERG and our instructors. This is our instructor team. I'm just the head instructor. I just coordinate people. I teach some of the wet lab content, as you'll see, but there are other people helping me teach this course who have better domain expertise. So first, I'm going to introduce Kat and Marius. So Kat was one of the originators of CSBERG, one of the early developers of CSBERG, and she has also helped develop a lot of the content to teach the DNA-related stuff. And so, unfortunately, I'll be taking over that, but Kat helped a lot with developing that material. Marius is our specialist, you could say, on protein, and so he'll be teaching some of that in the wet lab module. My dry lab folks are Braden, Kavya, and Tanyu, going over modeling, bioreactors, and machine learning. Some introductory material will be too intense, but to get you into the jargon and the field of bioreactors and machine learning, and really just generally speaking, biological modeling. S.A. Umar will be going over the human practices and talking about the social dimensions of synthetic biology. And lastly, we have Austin and Amy, who kind of bring everything together. So Austin is from OISE, the Ontario Institute of Studies in Education. He's training to be a teacher. And he's also one of my buddies from back in my time at iGEM Waterloo. And so he's helping bring in that theoretical framework for pedagogy and helping solidify a lot of our teaching pedagogy in CSBERG and helping us apply that to synthetic biology. And lastly, we have Amy, she was one of the presidents of iGEM Toronto, along with Umar and Kat. And so she has this amazing drive for leadership. So she'll be teaching some of the leadership stuff, not through SYNB0, kind of indirectly through our podcast series that we have elsewhere on our channel here. So I want to give a big disclaimer before I dive into teaching all this stuff with my instructors. SYNB0 is an introductory lecture. It's made for the novice synthetic biologist, and this is because it's an interdisciplinary field. There's a lot to cover, and what we want to do for the novice, for the rookie iGEM member, is to get them, to get their ball rolling, to get them with the jargon, to understand where they are in the field, and to understand where the field is going. And so, because engineering is this complex, multifaceted discipline that's tackling all sorts of issues, you have to bring in all sorts of disciplines to tackle these big issues: experimental, modeling, social sciences, as well as financial, entrepreneurial, political. There's all sorts of dimensions to synthetic biology. And so because there's so much to cover within one lecture series, we're going to focus on teaching the introductory material, the jargon, and the kind of feel for these different fields, so that you can later explore further through our courses and through our later lecture series, or through other online material. You can learn more specific topics there. So let's move to section B. What we want to do in this section is define what synthetic biology is, or at least come up with some working definition that we can use for the remainder of the SYNB0 lecture series. And so to kind of explain this figure a little bit, SynB0 can be defined through the lenses of other fields. So for example, what we often see are biomedical engineers providing a definition that has kind of a health lens into SynB0. Or you might see metabolic engineers who have a chemical manufacturing perspective giving a definition of SynB0 from their lens. And so in these definitions, they have these slight, you can call them flavors of SynB0. And what I really want to do is develop a working definition that is kind of free from these different lenses, And it's important to do this just so that you can broadly define synthetic biology, just because of how truly interdisciplinary it actually is. And so to help define synthetic biology, I go through its etymology, its connotation, so it's kind of, general understanding from society through kind of accepted understandings that people say on social media, for example. I want to define it through its history and how it developed into the field it is now, and define it through its purpose, what purpose it serves for society and for scientific research. And the meaning of synthetic biology is really important. It's not just a philosophical pursuit for the sake of semantics, of just philosophical arguing. It has issues in terms of legality, because when you start to develop policies and guidelines for SYNB0, especially at the international level, you kind of need a well understood and agreed upon definition that you can develop these policies and guidelines from. Now, the way I developed this working definition for SYNB0 is really inspired from Sophia Roost, who is this anthropologist at Harvard. And so she has written this book called Synthetic: How Life Got Made. I really recommend you read it. And what it really does is walk you through kind of the cultural development of synthetic biology. It doesn't dive into the technical detail very much. And it does that so that you can develop this kind of deep understanding of how the field is rooted and came to be to what it is right now. So in the first way to define synthetic biology, we want to break down the word itself. This is the etymology of the word. And so synthetic and the word biology, both of these words have Greek origins. Now, I'm not going to try to pronounce the Greek origin, just because my Greek friend will yell at me, Daphne. But for synthetic, it's a combination of the words, or of the phrases putting together and pertaining to. So pertaining to putting things together. And for biology, what we're probably more familiar with, it's the study of life. Pretty simple. And so just from the etymology, we can come up with a definition. Synthetic biology is the study of life, but specifically pertaining to putting it together. Doesn't matter if it's for fundamental or applied sciences, it's just putting it together. It's how you do that. So the next way to define synthetic biology is through connotations. Specifically, the feeling that is evoked when you say the word synthetic biology. What do you generally think of? What do people who identify as synthetic biologists typically describe it as? And so the origins of these definitions typically come from a place called Wikipedia, as you might have figured. And so in Wikipedia, the definition of synthetic biology is this area of research that is striving to create new biological parts, devices, systems, and redesigning them such that you can take nature's innovations and bring that to engineering. And so in Wikipedia, it kind of gives this big list of different disciplines that feed into synthetic biology. But what it is clearly missing are the social sciences. And that's a big flaw, and we'll explore that later. Another place that people get their definitions from, at least within the Canadian perspective, is through the Canada SYNB0 conference that we have each year since 2018. And so at this conference, they gave a definition that came from a dude named John Cumbers and Carl Schmider from SYNB0 Beta. And so from their book, this is the definition that they gave, that Canada SYNB0 also gave. It's the design of new biological parts, devices, and systems. You can kind of see a trend here. The redesign of these systems, and more succinctly in point C, it's the endeavor. Synthetic biology is the endeavor to make biological sciences more accessible to engineers, so that engineers can apply optimization algorithms, can design specific systems for purposes that are important in society. That is what synthetic biology is. It's making biology a more engineering discipline. And so putting these things together, these connotations, these general understandings, we could say that SYNB0 is a field where we treat biology as comprised of these parts and these systems, and by designing them in ways you can make it more accessible to engineers. So in this design process, what we want to do is make biology specifically more modular, robust. We want to simulate biology. We want to prototype it we want to make it more amenable to the engineering discipline based on these definitions through connotations. So the next way to define synthetic biology is through its history. Now, what I want to do is walk us through molecular biology and how it evolved into molecular biotechnology and later into synthetic biology. And I'm going to use the Pokemon metaphor for this with Pichu, Pikachu, and Raichu. But am I by no means saying that the early developments in molecular biology is baby work, because it's not baby work. I'm just talking about it in the context of it being early in the development of synthetic biology. So beginning in the 1950s, a lot of molecular biology was accelerated forward from the discovery of the DNA structure and the elucidation of its role in what we now know as central dogma, the flow of information from DNA to RNA to protein. So at this point, there's a lot of fundamental science going on, and a lot of it was beginning to occur at the molecular level, working with just the DNA, some proteins. And so moving forward a couple of decades into the beginnings of the 1970s, we saw the development of the first plasmid, the PBR322. And what plasmids allow scientists to do is to transfer genetic material between E. coli. And so you can transfer useful properties to E. coli and give them new properties that might allow them to do things you want them to do. And so with plasmids and with other biotechnological developments, we were able to develop some genome integration platforms that would allow for better agricultural developments and pharmaceutical developments. So this is where GMOs were beginning to form, and so crop GMOs. And this is where we started seeing some biotherapeutics being produced in the pharmaceutics industries by the production of protein therapeutics in mass, in bacteria, in bioreactors. And it was little bit later on where we started getting some developments in systems biology, which is really spurred through the development of Sanger DNA sequencing, which allowed us to read through DNA and understand the actual DNA sequence and kind of see where central dogma really starts from. And so in this phase of molecular biotechnology, we have fundamental and applied science at the molecular level, because now we can study DNA and proteins and RNA and apply these specific things to real-world applications. But we only really have fundamental science at the systems level, at the holistic understanding of the cell. But moving into the early 2000s, this is where synthetic biology really takes off. And so there are three technologies or developments that I think are kind of what drove home the development of synthetic biology. And so that's the ability to write DNA through DNA synthesis. Their chemistry is called phosphoramidite chemistry for DNA synthesis. The second thing is genetic circuits, the toggle switch, or also called the Collins switch from Jim Collins' lab at MIT, allowed cells to exist in two different states, and you could control which state based on the presence of a different chemical. And third, which deserves a mention of Drew Endy. So Drew Endy was originally a civil engineer, but he wanted to apply his engineering background to biological sciences from curiosity. And so in his PhD, what he was tasked to do was to model a phage genome and see how the phage genome could be modeled and then predicted to behave in a system. And so through his PhD, he found that it was insanely difficult to do this modeling. And so what most people who are trying to model biological systems would think is, let's just try to adjust the parameters, some fudge factors, to make the model behave like it would in real life. Now what Andy did, what his approach was completely the opposite. What he said was, all right, instead of trying to fix my model, how about I try to fix the phage genome? Because there are all these complexities with the genetic architecture. Let's fix all that, simplify it, so that it actually matches my model. And this was a driving principle behind the early days of synthetic biology. Rather than adjusting our models to fit life, how about we adjust life to fit our models instead? And so moving into now the mid-2000s, late 2000s, IGEM, the International Genetically Engineered Machine competition, which spurred from MIT, it grew rapidly. So it started with like a couple of teams, 12 to 13, mostly from MIT. and quickly emerged into hundreds of teams entering this competition from around the world. And this really spurred the development of new synthetic biologists and spurred investor interest, spurred government interest in this field. And so at this point in synthetic biology, when it's beginning to form, we have fundamental and applied sciences at both the molecular level and the systems level. So this is a history of how synthetic biology came to be. And what I want to do is to develop a working definition from this history. I'm going to introduce two timelines for synthetic biology that are published in literature. And from these two timelines, we can see how they're different. And because they're different, we can then develop the third working definition for synthetic biology. So the first timeline I want to introduce is from the Paulson Labs publication, Minimal Cells, Maximal Knowledge. And so in this timeline, what they do is look at the fundamental milestones in molecular biology and how that evolved into genomic and synthetic biology, how biology became an engineering discipline, but also in a very advanced fundamental discipline. So feel free to pause this video to look at these different milestones and come back to it. So what we can see is that there are some milestones here that are particularly pertinent for synthetic biology, and I want to point those out to you guys. So obviously the structure of DNA is kind of the starting point, in my opinion, that leads to developments in molecular biotechnology and synthetic biology. Also very important is the operon structure. So Jacob and Monod, they developed a mechanistic understanding of the lac operon in E. coli. And so this mechanistic understanding allowed us to better understand how genes are regulated, how transcription and translation are regulated by the cell. And you can imagine this has big implications in developing genetic circuits that control how the cells behave based off a couple of genes. Next, we have restriction enzymes, which basically allow you to cut DNA and stitch it back together. This was the molecular biologist's way back in the day to basically put DNA together to do recombinant DNA technology, as you might have heard. Next, we have DNA Sanger sequencing. So by being able to read DNA, we can define exactly what a gene is made of, what the DNA segment is. And you can imagine how this has implications in disease, because you can check and compare the gene from one healthy patient to a sick patient. What you can also do with sequencing, obviously, is to enable genetic engineer. Because we know this particular gene, which is encoded by this DNA sequence, produces, say, insulin, for example. Now, if we transfer a plasmid containing a gene encoding for insulin into E. coli, theoretically, you could mass produce insulin, which is what has been done. Next, we have polymerase chain reaction, or PCR for short. Typically, when you start working with DNA, you want to have a lot of it to work with, some manageable quantity. And so sometimes when you're working with genes directly from microorganisms that don't have a lot of biomass, you won't be able to get a lot of gene to work with, and so you need PCR to amplify it. Now PCR has developed insanely. There's all sorts of modifications you can do to PCR to allow for different assembly methods. And of course, PCR is also used in forensics and diagnostics. Linear programming for maximal theoretical yield is the basis for flux balance analysis, and it's what metabolic engineers use to determine how different deletions and introductions of new genes will affect the metabolites in the cell. And so what I previously just described, so that timeline really focused on the fundamental discoveries leading up to where we are now with synthetic biology. The second timeline I want to introduce is from the Collins Lab and their publication, A Brief History of Synthetic Biology. Now there's two parts of this timeline. I'll show the first half. And so feel free again to take a pause and come back to this video after you look through the different milestones in this timeline that are highlighted. Okay, so hopefully you've looked at it. I want to highlight some of these. So as I mentioned before, the lac operon. Understand the mechanistics behind transcription and translation, and how that mechanistic insight affects this gene operon, this gene structure. So again, that's shown here in this timeline. Of course, kind of summarizing everything is the development of molecular cloning techniques. So restriction enzymes is one of them, PCR is one of them. DNA sequencing is also part of this. This is really big. And so, which is one part that's very different from the previous timeline, is the mention of the toggle switch and the repressilator. So these are two different genetic circuits that have different functions. But what they do is they build on our previous understanding of genetic regulation, and they apply it so that you can control cells in two different states with chemicals, or you can cause two different genes to repress each other over and over again in a cycle. And so this is the first emergence of genetic circuits and kind of spurred synthetic biology onward. As I mentioned before earlier on in this lecture, the development of the Artemisinin project comes from the development of this pathway to produce metabolites in the Artemisinin pathway in E. coli so that you can mass produce it in big bioreactors. And so this is a Kiesling effort from the Kiesling lab. And so this is the second half of that timeline from before, from the Collins lab. And so again, I encourage you to take a pause in this video and look at these different milestones. So obviously these are all very important. I want to point out around 2009 is when we saw the development of the Gibson assembly. Now, a lot of people would be like, okay, boomer for me when I say this, but before you use Gibson assembly, which is this one-pot reaction to combine all your DNA together, learn restriction enzymes and how to use them, because it's kind of like your scissors. You want to learn how to use scissors before you use anything more complex. But anyway, the Gibson DNA assembly is one of the big things that was produced that people typically use nowadays for DNA assembly. And some other important things are continued developments in switches. Switches referring to the toggle switch before, but also more broadly, the continuing development of genetic circuits to be able to control cells more logically, more digitally, in a more analog fashion, so that it's more amenable to engineering. And lastly, very importantly, so we saw that the Artemisinin work started much earlier on in the 2000s, and over the years, it was constantly developed, constantly revamped, to the point where we had an engineered yeast strain that was able to express the necessary pathway to produce artemisinin at an industrial level. And so Sanofi, the pharmaceutical company, was working with Amyris, the company that was created by Kiesling's lab and Jay Kiesling, and they basically scaled up this entire bench scale work to industrial scale to produce literal tons of artemisinin to sell to different countries to help deal with malaria. And so in this particular timeline, what it really focuses on is the applications of molecular biology techniques, of using those techniques and our understanding of DNA sequencing and DNA synthesis to derive forward new applications. And so from this, we can kind of develop a working definition for definition number 3. So to get to the third working definition, we can highlight the differences between these timelines. So for Paulson's timeline, it really focuses on the early developments of synthetic biology, or just general biological engineering, and also the aspects of synthetic cells, as you saw. For the Collins timeline, it really focused on the genetic tools, so the switches, the circuits, and their applications. And so to summarize it in a different way, for the Paulson timeline, it really highlights the fundamental basis of SYNB0, the bottom-up approach. Whereas for the Collins timeline, it really highlights the applied basis of synthetic biology for the top-down approach, identifying what you want the synthetic biology to be used for, and then building your way that way. And so for the bottom-up approach, to kind of put this more simply, when you put life together, you are able to understand it a lot better. This is something called maker's knowledge. You know, as an analogy, if you're trying to create pasta, you can create pasta by analytically studying how spaghetti expands, how pasta sauce can be cooked. But really, you just want to go ahead and just make it. And by making it, going through trial and error, you learn more about spaghetti. And so with life, by putting it together, you can better understand its mechanistic, its fundamental aspects. And so this push towards developing synthetic cells, which is a big part of synthetic biology nowadays, it's developing these minimalist cells, these synthetic chromosomes, that allows us to better understand DNA, understand genomes, proteins, these systems of biological entities, and how that relates to cell viability. Understanding this by creating cells is one part of synthetic biology. Now, in the Collins timeline, it's a little bit different. So by putting life together, what it allows us to do is to use it for better purposes, environmental or human purposes. And so genetic tools allow us to manipulate cells so that you can control them for the early uses of pharmaceuticals and agricultural uses, and more into this century, the industrial and the information process applications. And so when I say information processing application, I'm alluding to genetic circuits and using them in logic gates to process information. And what I want to also point out is underpinning both this fundamental basis and its applied basis is DNA synthesis and sequencing. And additionally, rewriting the DNA through editing. And so CRISPR technology plays a big part of this. And so what this slide is doing is giving you a big overview of what synthetic biology is, the fundamental side, the applied side, and the technologies for DNA for writing it, for reading it, and for editing it that allow us to manipulate these parts, these systems, to design our biological inventions. So let's kind of bring everything together. So far I've defined synthetic biology through its etymology. So from the etymology, we understand that SYNB0 is the study of life pertaining to putting it together. And I also defined it through its connotation. So people generally understand SYNB0 as this field where we treat biology as comprised of parts, of systems, things that we can design. And in designing it, we make it more accessible to engineers. We give it properties like modularity, like standardization, such as simulation as well. We make it more amenable to engineering design. And in the third definition, we looked at the history of synthetic biology, those two timelines. And from this, we understood that SYNB0 is both a fundamental science and an applied science, meaning that it's engineering. And by using both of these different approaches, you can use SYNB0 to explore the truths about life, the fundamental curiosity-driven approach, and also use it to design new systems that are biological in nature for the betterment of us, of everyone around us, and the environment as well. And now I want you to take a pause a little bit here and stop here a little. What I've been trying to do is talk about synthetic biology in all of its good. I've been talking about how it can benefit the world typically. And I wonder if you can see where I'm going with this, because although you can use it for the good, if we look to the media and how it portrays biotechnological innovations in the future, typically it's more dramatic to talk about the negatives that can come about from bioengineering. And so if you look to the TV shows and the movies and even in anime, you can see how biology and engineering it can lead to some negative consequences, some things that you may not want in some unethical situations. And so what's really important in defining synthetic biology is defining it through what it is intended to be used for, its purpose. This is getting to the fourth way to define synthetic biology. And so before I move into that, to kind of bring these three definitions together, I've kind of formulated this definition here. This is taking me actually a couple of years to get to this definition. And so synthetic biology is an interdisciplinary field. Similar to cognitive science, it's an interdisciplinary field comprised of different subdisciplines coming together. And it's about creating life, specifically these living robots, you can think of it, both the molecular machinery and the cellular machinery, and using these systems so that you can start developing characterized parts that are standardized, characterized systems that are able to do a defined set of tasks. And by creating these machines, these living robots, we're able to better understand life, but we're also better able to create things that are useful for the world. So synthetic biology is very akin, so I mentioned cognitive science before. It's also very akin to mechatronics engineering. And so mechatronics engineering dates back to a fundamental period of the development of calculus, the same way synthetic biology dates back to basic developments in molecular biology. So if you look at this particular figure, that kind of shows how mechatronics exists and what it is comprised of. We can see that mechatronics is comprised of electronic systems and mechanical systems working together by using computers and control systems to make that integration happen. And by integrating these four different circles, you get different technologies that exist. And with these technologies, what you're able to do is then apply to all sorts of industries, all sorts of fields, to allow humans to have access to much more advanced technologies. And so mechatronics is a real conglomeration, a real aggregation of all sorts of knowledge. So if we accept this notion that synthetic biology is ultimately going to be an engineering discipline, if it isn't already an engineering discipline, we can start thinking about how synthetic biology will grow and how that affects the definition. So if you look to traditional engineering disciplines, we very quickly see that some engineering programs have specializations dedicated to look into how engineering affects society, the ethical and the global implications. And of course, we know that engineering also has financial and commercial implications as well. And so what I'm talking about more specifically is in some engineering programs, they have specializations specifically for engineering in society. And there's also engineering economics and entrepreneurship. Now, synthetic biology, if it's an engineering discipline, these are two dimensions that it has space to still grow in, has space to still expand into. And so I want to focus a bit more on the social dimensions of synthetic biology in SYNB0 through Estee and Umar's work and instruction. I'm going to focus less, in fact, I won't focus on the entrepreneurial and the economics of synthetic biology, just because we have partners in the future who will provide this content through their channel and through their group. So look forward to that through CSBERG and through our Twitter account. But for SYNB0, what we're going to focus on is the social dimensions of synthetic biology. So let's try to define synthetic biology through its fourth approach. And so what we're going to look into is how to define synthetic biology through its purpose. Now, I said before that SYNB0 is a means to study and design life, which is both going to be a fundamental approach to discover truths, but also to benefit society and people and the environment, which is the applied approach. So these are its intended purposes. We intend to use synthetic biology for the greater good. So let's take a look at one example, a little mini case study. Monsanto, through the 1950s into 2000s, benefited greatly from advancements in molecular biotechnology. And they were able to develop crops that had better properties compared to the wild type, the natural version. And so these GM crops, these genetically modified crops, specifically they were developed to help stabilize food production in certain areas, to increase crop yields because they were more resistant to the environment, maybe improve farmer livelihoods because they could have better stable income, and provide consumers theoretically with higher quality food that is more nutritious, just larger fruits, for example. These were the intent that Monsanto had. And what we found was that biotechnology did such a great job for so many crops that it offered this massive advantage over organic crops. And it was incredibly lucrative. And because it was so lucrative, it led to a lot of what I say multinational high-profile lawsuits. You can Wikipedia Monsanto and you can very quickly see a big summary of them. And a lot of these issues had to deal with the fact that there was a lot of monopolization due to this conferred advantage. And there was also some ethics behind the testing that were unclear. The long-term effects were not as well studied or at least communicated to society, to the public, where they're major stakeholders in this because they're the end consumer. They will eat these products, but they weren't communicated with in regards to how this was developed, this technology. And so if you look through and search GMO on Google Images, you will see that Monsanto basically single-handedly altered the perception of molecular biotechnology because there was so much angst and anxiety, fear, mongering, and miscommunication between the science that was going on in Monsanto and the intended good purposes of it, but also the implication of how the business affected the communication with the stakeholders, the end consumers who are eating this product. So in this next example, this is the second mini case study I want to show. Again, the intended purposes of synthetic biology is typically to discover truths and to apply it to society to address some problems. Now, if we look, you've probably heard of the whole CRISPR baby situation over in China, and if you haven't, definitely Google it. Just type in CRISPR baby. And so the idea behind this research that was going on in China was to help make humans become less susceptible to becoming infected by HIV and therefore developing AIDS. And so the idea was to use CRISPR gene editing technology to modify genes in the human genome that would make us less susceptible. That was the whole purpose, to kind of basically mitigate the spread of AIDS. Sorry, the spread of HIV to cause AIDS. And so what happened was they were editing some human embryos, but the issue was that these embryos, they were never at risk of contracting HIV in the first place. They weren't susceptible to it. But what we did was we introduced these kind of the gene editing technology to modify these genes. And the issue with CRISPR is that it still has unintended off-target effects that can lead to other mutations, that can lead to other diseases that you can't immediately detect with the baby. You might not be able to, but you may detect it much later on in life, and that affects their quality of life, these babies. And the babies are actually born too. They weren't euthanized. The babies are born. And this is another layer of ethical implications behind CRISPR technology. And so what happened was the whole entire CRISPR community internationally was very much on the brink of holding this moratorium, which is a word to describe the stopping, the arrestment of all activity related to research on CRISPR. And that was because scientists felt like this is not a good idea to gene edit a human embryo and let it be born afterwards. And so we needed to develop policies to kind of regulate scientists in this field at an international level. Now again, the intended purpose was to help prevent the spread of HIV, to minimize suffering through AIDS, but the communication from the scientific community to the public was not really there. So based off of these two case studies, we can very quickly see that molecular biotechnology has been very heavily criticized, particularly due to the lack of scientific communication, which really affected the way the public perceived the research that these scientists were doing, these molecular biotechnologists, and which negatively impacted the acceptance of the field, the acceptance of GMO crops, despite the good that they do provide for society. Now, molecular biotechnology is the predecessor to synthetic biology. We mentioned this in the history of synthetic biology. And what synthetic biologists nowadays have learned from is the mistakes of the past. Synthetic biologists now are much more proactive and involved with policy development, with the governance of the field, and with surveilling biosecurity in the world. And so if the purpose of SYNB0 is to do good for us and our environment, there has to be some kind of judgment process involved in this, because good and bad they can become very subjective based on people's personal agendas. So there has to be governing bodies to look after the field of synthetic biology, to support it, to regulate it, such that the good intent is what prevails and not the bad intent. So what's really important that synthetic biologists have acknowledged is that a synthetic biologist isn't just someone who has technical expertise in terms of the experimental and the modeling. It's also someone who has very strong and deep understanding of the social sciences and the ethics. And these are typically people who are social scientists and ethicists with specific training in these disciplines, who help bring their insight into science communication, into public perception and acceptance, into the design cycle that engineers and biologists and synthetic biologists use to build these living robots. So to kind of phrase that more simply, we have these social scientists and ethicists as a part of the entire design cycle, as a part of the process of making these useful machines for our society and for the environment. And so from people who are watching this video who are researchers, undergraduate, graduate, postdoc, who are researchers at universities, really what you really think of is university research and maybe the private public sector if we're talking about funding. But what's really happening, going on in the background to bring technologies into society are all sorts of stakeholders. Stakeholders is a key word that you want to think about when you think about human practices. And so in this figure, I would encourage you to pause the video and see what the relationships are between these different stakeholders and how our small world of university research, maybe through funding through the private and public sector, is actually just a small part of the bigger map that is society, regulation, governance, and surveillance. Okay, so this is the final slide for section B. But what I want to do is bring together all these definitions that we've come up with to define synthetic biology, and then kind of synthesize a working definition we could use for SYNB0 that I encourage you to also use in your own practice and exploration of synthetic biology. And so we first define it through its etymology by breaking down the word. And through that, we understand that SYNB0 is the study of life, pertaining to putting life back together into one working part. And then through defining SYNB0 through its connotations, we understand that most people generally understand SYNB0 as this field where we treat biology as this orchestra of parts and systems that comprise living systems. But these parts and systems are things that you can design in such a way where you make them more modular, where you standardize them, you better characterize them. And this allows it to be more amenable to the engineering design process and more accessible to engineers. And we also define synthetic biology by looking back into its history. And we saw by looking at two timelines and defining what the differences are between these timelines, we saw that synthetic biology is a fundamental and an applied science. And by wielding synthetic biology, you can better understand the truths of life by putting it together. And by putting it together, you can also use it to create new systems that don't exist in nature, that have applications that improve the human condition, that improve societies and the environment as well. And so when I combine these three definitions into one, I introduced this definition. And the issue with this definition is that it's lacking the social dimension that is really important to synthetic biology. And so by looking at those two case studies we just talked about and putting those together, we find that integrated into the design process of making these living robots, integrated into the commercialization of these creations, is the need for social sciences and ethics. And the need for them is so that we can uphold these standards and values to develop policies, to ensure that biosecurity is being done by these governing bodies, and to promote innovation safely so that the greater good prevails and the unintended purposes are minimized and mitigated. And so this is the working definition I want us to use moving forward through SYNB0 as you look through other lectures. I encourage you to pause here and take a deep look at this definition and internalize it so that you can kind of contextualize the wet lab, the dry lab, and the human practices modules all together into one more cohesive understanding and a holistic understanding of synthetic biology. So now I am going to move on to section C. What I'm going to do is take a little break first. I'm going to tell you a little bit about my experiences working with iGEM and just for fun, so you can take a break here, maybe listen to me, and then we'll move back into section C, where we will talk about the Artemisinin project. So hopefully you're taking a break right now. These lectures can get pretty long, but we try to make them not too long so that they're within what you would encounter in real life in lectures, so under 2 hours at least. Well, anyway, right now I want to take a little break and just tell you two stories that I have from my iGEM experience. I have a lot of stories that you'll encounter throughout my lectures at least in SYNB0. But for right now, so the first story I want to tell is this time I had in Boston. So this is my second time going to Boston, and I was on the iGEM Waterloo team, and I was one of the wet lab leads at the time with my co-lead Alicia. Um, Hello, by the way, if you're watching this. Uh, I don't know if I've told this story to anyone in the iGEM Waterloo team. I may have. So there was, I think it was the night where we had just presented to the panel of judges our entire presentation for the work we were doing the entire year, and I thought it went pretty well, and I was ready to celebrate. So what as we university students do is get alcohol from somewhere, and so in Boston they don't have LCBO like they do in Ontario. Um, they have independent liquor stores, and they do, and so we were basically looking for alcohol. We were thirsty. I think at some point I had picked up a sake that I cannot find in Canada, and with the sake I had obtained a bucket of ice to put it in. And all I remember from this memory, you'll see why this is all I remember. So I was in the hotel room of one of my wet lab members, and they were about to go out. I wasn't sure what they were going to do, but for some reason, I was left in the room by myself, and I had this bottle of sake in this bucket of ice, and I decided, hey, you know what? They're off to have fun. I led this team for this past year. Let's take a little break now and relax now that we're done the presentation. So as I do, I drank the sake, and I drank a lot. And at some point, I think I lost track of time, but I was one of I can't say their names. One of the members walked into the room and saw me drinking the sake by myself, looking, I guess, mystically out the window. This is the view that I had, and I was looking out this window, and he was, obviously expressing concern, making sure that I was okay. Uh, the fearless leader better not be about to die on him. And so, I said things were fine. Okay, this is where I lost my memory. So at this point, based on the pictures I took, because I took my camera with me to take pictures as I was drunk, I was basically wandering Boston, and somehow I ended up on their transit system, and I took the train. Again, I'm entirely drunk at this point. I'm taking pictures along the way. Uh, I'm taking pictures, and I'm drunk. I'm on the subway, and eventually sunset falls, night hits, and I'm leaving the subway station that I get off at. I'm still like pretty buzzed, and I'm not sure where I'm going. And I walk out, and I see a burger joint called Tasty Burger. So so I've been in Boston 5 times now. I always visit Tasty Burger because of this memory. I see a Tasty Burger, and I go into it. I order this burger, and that's why I have this amazing picture of this burger over here. Um, I'm totally drunk out of my mind. So I have this burger, and then what I think happens is I fall asleep. I wake up, and it turns out it's like 2 a.m. at night, and I'm in the middle. What I realized was I was at Harvard at the Tasty Burger nearby eating a burger, drunk. I wake up and I'm like, oh God, what am I doing? And no one knows where I am. No one's even messaging me. So I assumed, okay, you know what? No one really cares. I'm okay right now. I've woken up. And so I decided to just walk back to the hotel back near the Heinz Convention Center. So I'm walking from Harvard back towards MIT across that bridge. And this is, my last season being a member on the actual team that's competing. And this was just a walk that I uh, am very nostalgic about, because it was during this walk that I reflected on, what do I want to do with synthetic biology. A lot of people who go through the IGEM competition typically don't stick with synthetic biology, which is fine. It's an opportunity for you to do research, right? But I asked myself, what do I want out of synthetic biology? And this walk I had, I was relatively sober at this point, after that burger. It was at this point in this walk that I kind of realized that I really wanted to commit myself to the whole synthetic biology journey. Um, and that's why I've been doing my PhD research on stuff related to synthetic biology. So fundamental research that I want to bring into the limelight of synthetic biology, has to do with metal recovery. Um, and so I really devoted myself to synthetic biology, and now I'm teaching it, I'm the biggest nerd possible. Um, I'm trying to teach this material so that people can see how useful this field is, so they can bring their ideas that I can't even imagine, and bring them to the field of synthetic biology and make them real, make them feasible, something that can actually serve society. Because what happens a lot in iGEM is you have these crazy ideas, people do some preliminary work to show that it might work, some proof of concept, but it's never followed through. And so what I want to do is create some material to help teams start off with like hitting the ground running is what they would say, um, so that they're able to spend more time thinking about the translation of their technology into actual practice. And of course, it's only a small part of it. It's not the solution. CSBERG is but one part in this whole journey. But yeah, that's my story of my drunken burger situation back when I was doing iGEM. And so the other story I want to tell is a much shorter story. So this story happens before the whole Boston situation I just talked about. This is like a month prior. So I was in the midst of entering my 5th year. At Waterloo we have co-op, so I have 5 years because we're delayed because of co-op. And so I was entering my 5th year. I was moving into my new place, my new apartment called Icon in Waterloo. And Waterloo apartments are notorious for not being ready for students to come in uh, on the dates that they're supposed to come in because they're still doing construction. So Icon was no exception, and for a solid week, I had no place to live. And where else could I live besides the iGEM office that we had for the Waterloo team? And so in this picture, this is my office space, dedicated just to me. There's one corner of this table dedicated just to me, so I can basically live here. And what you don't see is to the left of this picture is a little space where chairs are. I actually slept there. I literally slept in this office for an entire week. I don't think anyone actually knew because I hid my sleeping bag, but I basically lived there, and I would shower in the engineering building just down the road, and it was a weird time. iGEM, I think many people, especially those who are watching this who are from iGEM, can have their own very unique experiences. This is my unique experience of iGEM deeply impacting my life, but this is how iGEM supports students through research and through life. So those are kind of my 2 stories I wanted to share. There's more stories in the future. To kind of wrap it up, so what we're going to do now is switch from section B, where we define synthetic biology, and now move towards talking about one of the biggest synthetic biology success stories, or seemingly success stories, as we will see. And it's something that a lot of synthetic biologists will refer to. So it's good that you know what this whole story is, what this whole project is. And then after that, I'll talk about what's going to happen after this lecture, what you can look forward to. So without further ado, let's move on to section C. So for section C, we're going to focus on this paper, Semisynthetic Artemisinin, a model for the use of synthetic biology in pharmaceutical development. So this paper is published by Chris Padden and Jay Kiesling, and what they do in this paper is summarize a decade's worth of research that has gone to develop a yeast strain that's been engineered to produce the precursors for artemisinin, so amorphidiene and artemisinic acid. These are the things they're producing in the yeast strain to a specific titer, and what they're trying to do is basically produce it so that you can then later semisynthetically convert artemisinic acid into artemisinin, the actual antimalarial drug. And so what we're going to do with this paper is try to understand what synthetic biology looks like in practice. And so what I'm going to show is that synthetic biology draws on many fields. It's an interdisciplinary field. And one of the fields it draws on is this whole systems biology and metabolic engineering discipline. And these are well-established fields already before synthetic biology. And so what I don't want to kind of come off as is by trying to say that SYNB0 is this umbrella field where all these different disciplines that it draws on are under its umbrella. I don't want to say that synthetic biology is its own thing, but it draws on the knowledge from other disciplines. And so there is no perfect example of SYNB0 just because of how interdisciplinary it is. But this example here with the Artemisinin project is a clear example of what synthetic biology could look like. So the Artemisinin project was developed and funded by the Bill Gates Foundation and a company called Path Drug Solutions, as well as the University of Berkeley and the Kiesling Lab. And what they're trying to do is to be able to create a sustainable source of artemisinin to deal with malaria that is endemic to different countries across the world. And these are typically going to be countries that are not as developed, and so they don't have easy access to these kinds of drugs. So here is a map of where malaria is endemic. In particular, the turquoise is a statistic for the 2015 year. And you can see how widespread it is across many of the developing nations. So malaria is spread by mosquitoes, and it affects millions of people. The best way to treat it is through a drug called ACTs, which is short for antimalarial combination therapies. And so the primary ingredient for ACTs is artemisinin. Now, a Chinese researcher and Nobel laureate named Tu Youyou, she basically discovered artemisinin. And it turns out that artemisinin is from a plant called Artemisia annua, which was previously used in traditional Chinese medicine way back in the past to treat malarial symptoms. And so by discovering what artemisinin actually looks like structurally as well, we were able to identify exactly what was needed to treat malaria. But the issue is that when you want to produce drugs from plants, you have to deal with the fact that you need to grow the plant. And so as any farmer can tell you, sometimes crop production is very dependent on the weather, the general climate. And so there can be very unstable production of these plants that can lead to huge price fluctuations in the actual downstream drug, and in this case, ACTs. And so this has a direct impact on patients and their pocket, and it also has a direct impact on governments who are giving subsidies to buy these drugs for their populations. And so generally speaking, the price of ACTs was fluctuating quite largely. And so Jay Kiesling is a professor at UC Berkeley who spearheaded an initiative to find a solution to deal with this unstable production of artemisinin, which could then lead to better supply of this drug to people around the world who are affected by malaria. So I just want to point out real quickly, Jay Kiesling his lab has produced a number of very successful professors. I just want to point out Michelle Chang, Christella Prather, and also a Canadian, Dr. Vincent Martin as well. All of these doctors, they come from Kiesling's lab, and so we'll talk about a bit about them more in the future, but just wanted to point that out. So Kiesling has formed a company called Amyris, and the company is funded by the Bill and Melinda Gates Foundation to develop some sort of production line to semi-synthetically create artemisinin using genetically engineered microorganisms or machines, and this is what GEMS stand for. And so Kiesling worked. Oh, I forgot to mention, if you've noticed that Kiesling looks like he's pretty buff, it's because he was actually a college wrestler at some point. So yes, he is actually jacked. But anyway, Kiesling worked with a dude named Chris Patton to really help push forward this artemisinin project through Amyris. So before we talk about the metabolic engineering to produce artemisinin from these microorganisms, it's helpful to talk about the natural biosynthesis of artemisinin. And like I said before, engineering is based off of science, of our fundamental exploration of the world. And so synthetic biology is based off this fundamental exploration of molecular biology, and in this case, the biology of the biosynthetic production of artemisinin. So what we have here on the left side is a depiction of a plant cell of Artemisia annua intaking CO2 into the chloroplast and through its own metabolic pathways producing acetyl-CoA. Now plants have a lot of different secondary metabolites, and so FPP is the first committed step into producing a bunch of different isoprenoid precursors. And so FPP is formed, and then the first committed step towards producing artemisinin is by producing this compound called amorphodiene. So amorphodiene goes through a series of oxidations to get to artemisinic acid and dihydroartemisinic acid. And in the plant, what happens is that it will then be converted into artemisinin using oxidation and sunlight. Now, it's not exactly clear how exactly this happens. There's a process called Hawk fragmentation that might be the basis for this. But generally speaking, dihydroartemisinic acid is converted to the actual final drug that we use for ACTs through oxidation by O2 and sunlight. And for a more specific look at this whole metabolic pathway, here's one organic chemical reaction pathway where we start with FPP, and through a cyclization process made possible by this double phosphate group, we get to amorphidiene. And next, this carbon at the bottom of amorphidiene is then oxidized in different ways, and ideally it'll get to the point where it gets to artemisinic acid, which is the same thing here. So artemisinic acid can be converted to its dihydro form here through an oxidation again. And then again, through a series of bunch of other oxidations, it will get to the final point of artemisinin. Now, for Kiesling and Amherst to actually get a commercially viable pathway working, they need to get at least 25 grams per liter of amorphidiene, or at least 25 grams per liter of artemisinin. These are the kind of their two goals they were juggling as they developed through the decade. And so our target is to produce this much of artemisinin, and I'm going to use these values as a representation of the progress they're making as they use different approaches to develop this pathway. So to create FPP, which will then be later converted into artemisinin, there's a lot of stuff that goes on beforehand. And so in E. coli, they have the DXP pathway, and in yeast, they have the mevalonate pathway to produce FPP, which is then later to convert it into C15 isoprenoids. And C15 means compounds or metabolites that have 15 carbons in them. So their first approach, early in the 2000s, was to overexpress some of the enzymes that would create these intermediates. And specifically, they would express the rate-limiting steps. So to get the flux being pushed through from glucose all the way into this path and producing FPP. So when they were doing this, they only were able to obtain low milligram per liter, and what they needed, again, is 25 grams per liter of amorphidiene. So moving forward, they took approach number 2, and what they're going to do now is take some of these mevalonate pathway enzymes and put them into E. coli, and they'll take one plant enzyme and also put it into E. coli. And the reason why they want to use E. coli is just because it kind of grows really fast. It has one of the shortest doubling times compared to a yeast. And so if you can mass produce E. coli, you have a faster production rate. And so they put these yeast and plant genes into E. coli, and they overexpressed them, and what they were able to get was 0.5 grams per liter of amorphidiene. They then did some further studies in approach number 3, where they basically did some engineering, some protein engineering to these proteins, and they also took MVA and MVAS from Staphylococcus aureus as opposed to yeast, which was originally used, and they put those ones into E. coli. So now you have genes from 3 different organisms in E. coli trying to produce amorphidiene. And so through further optimizations in the fermentation process, they were actually able to get over 25 grams per liter of amorphidiene. Now, if you look back to that organic chemistry reaction pathway, amorphidiene is here. And for it to get converted into artemisinic acid, which is a closer precursor to artemisinin, it has to go through these oxidation steps. And so to engineer the yeast to do this, you have to introduce some enzyme that's going to allow for this specific oxidation path. So essentially, in a lot of microorganisms, cytochrome P450 is used to oxidize different metabolites and compounds. And so we needed a cytochrome P450 to oxidize amorphidiene into artemisinic acid. But the issue is that E. coli is not very good at expressing eukaryotic monooxygenases, or cytochrome P450s. And so at this point, what they said was that they had a quandary. They had an issue. Do they stick to using E. coli that could produce 25 grams per liter of amorphidiene, which is only this step here? Or do they look at using something that's eukaryotic, like yeast, that is able to actually use the cytochrome P450 to oxidize all the way to artemisinic acid. And so this is a point where I want to point out that in synthetic biology, when you're working with the wet lab and you're trying to determine which microorganism to use, what your chassis is to do your prototyping, it's very important to consider the chassis, because model organisms will allow you to do some basic prototyping just because they had the most studies done on them through molecular biology. But it's also important to consider industrial strains that may not have experienced as much scientific rigor and criticism and research, but industrial strains that are more amenable to large-scale fermentation processes. And so in approach number 4, they found a cytochrome P450 called CYP71AV1 and CPR1. And so what they're able to do is express both in yeast, and they found that it could produce artemisinic acid, but it couldn't produce enough. So what I also want to mention is that for approach number 4, there's a difference in the temperatures that the E. coli and the yeast were able to produce the artemisinic acid. And so this difference in temperature is really important, because in a large-scale fermentation process, any energy input you put into like a 200-liter reactor or even larger, it's going to have a significant, meaningful dollar value to it, which really will affect the downstream price of the final product. And so in fermentation processes, it's very normal to see that the system heats up. And so to have to cool the system down to 20 degrees might have a large cost to that, which will affect the pricing. And so it's important to consider whether you can produce this pathway to produce this final product at temperatures more suitable for fermentation processes. But anyway, what we have here in this slide, so what we see are the pathways that, or the enzymes that comprise the pathway that are going to convert acetyl-CoA in Saccharomyces cerevisiae all the way down into artemisinic alcohol. And so when they put the system into a strain called SYNB0, which is an industrial strain, versus their previous version, which was with the model organism S2A8C. With the SYNB0 strain, they were able to produce 40 grams per liter of amorphodiene, which is really great. But the issue is that you need 25 grams per liter of artemisinic acid, and through this whole pathway, they actually weren't able to produce enough artemisinic acid. There's basically a bottleneck at the point past amorphodiene. So if you look a little bit further down the process, so we have amorphodiene here, being converted to artemisinin eventually. Now, what they discovered through just fundamental research are the enzymes ADH1 and LDH1 from the plant itself. And it turns out that it took 6 years for them to find these enzymes that have this specific role of oxidizing and converting artemisinic alcohol and amorphodiene into artemisinic acid, which is the direct precursor to artemisinin. So it took a couple of years for researchers and scientists. I don't think they were from the Kiesling lab to find these enzymes, and then for the Kiesling lab to find out that these exist and to actually deploy them in the yeast strain. So eventually, what they're able to do by implementing these enzymes and by further optimization of the fermentation process is get 25 grams per liter of artemisinic acid, which is this thing right here. And that's what they need in order to go through the synthetic chemical conversion into artemisinin. And so that's why it's called a semi-synthetic pathway or process, because biologically we convert glucose all the way to artemisinic acid through this engineered pathway in yeast. And once we have artemisinic acid, which is released into the culture, into the broth that the yeast are growing in, it's after it gets there where it precipitates, and we can then isolate that, purify the artemisinic acid, and then chemically convert it through something called a Hock fragmentation in the presence of oxygen. And what it's basically going to do is just oxidize this particular part of the ring and introduce this epoxide group. And so there you have it. So they're able to produce artemisinic acid at a decent economical level, produce artemisinin. And so what they're going to do next is work with, or what they have already done, is work with Sanofi to to upscale this fermentation process with the yeast to produce artemisinic acid biologically. And Sanofi will provide some of the technology to do this chemical conversion synthetically to get to artemisinin, which Sanofi will then produce and turn into the ACT drug that's needed to treat malaria. So what I want to do is offer some perspective on the Artemisinin project. And so the Artemisinin project, or even the longer version, the semi-synthetic Artemisinin project, it was clearly a success in terms of it produced a living biofactory that was commercially viable. It could produce 25 grams per liter of artemisinic acid, which is what they needed, and they knew that at the beginning of the whole process. Now, it's clearly a success on that front. But what I want to do is offer a bit of criticism, or at least some alternative perspectives on what they could have done better to improve this process, or at least how other people in the future who want to produce metabolic pathways and engineer them should consider. And so what they didn't benefit from in doing this decade-long marathon of research were advances in computational strategies, as well as dynamic modeling of biological systems. What I'm trying to say here is they didn't use computer simulations to help them predict how these new enzymes might affect the entire, what we call flux, the flow of metabolites from glucose, the starting material, down to artemisinic acid. They didn't use that. And they really could have used that, but it might be because the strategies back in the early 2000s or the mid 2000s weren't as mature or developed. And so they may not have been as readily available to the Keesing lab. But definitely in the future, for teams, for people who want to do metabolic engineering, the computational path is typically the go-to to do it more rationally. What they also didn't benefit from was protein engineering, the advances in it. So they did some protein truncation to kind of limit the toxicity of some of their proteins, or some of their enzymes. But there are way more advances in protein engineering in the directed evolution space, in the de novo design space, that would allow them to develop better enzymes to produce their products at higher rates. I'll talk about that in a bit. So like what we saw in the Artemisinin project, whenever you rewire metabolism, like you introduce new enzymes or maybe you delete some stuff, or whenever you kind of change the central dogma of the system, or not change it, but like modify how it works a little bit, we enter the field of metabolic engineering and this entire massive realm of kinetic modeling. So more specifically, flux balance analysis and linear programming, or FBA and LP for short, these are computational algorithms that let you predict how metabolites are flowing through the cell over time and how changes in different reactions might affect the entire overall phenotype of your cell, such as the growth rate, for example, or how it uses up glucose and oxygen and other parameters. And so this idea of computationally modeling these organisms and predicting their behavior from these reactions has been advanced through dynamic control and through strain optimization. And so this is one picture that you can take a pause and look at this. You can compare these two to see how you can do the dynamic control. As well, this is another figure you can take a pause to kind of look through. And so these figures are from a recent literature review by the Mahadevan Lab. And what it summarizes are these advances in simulating the cells and their production of these compounds. And so underlying, when I talk about the central dogma and repurposing it, or at least like changing and modeling it, kinetic modeling is really helpful in this situation. And kinetic modeling is typically based off of ODEs and PDEs, which is short for ordinary differential equations. As you can see here, this is a system of ODEs, or more complex partial differential equations. But typically ODEs is what you need for most simple applications. And so it's by using these ODEs where you can model the cell and how these different macromolecules will interact with each other over time. And also importantly is stochastic modeling. Now, this is an area where Braden will help you kind of explore into, and it talks about how biological noise can be kind of categorized into extrinsic noise and intrinsic noise. And this categorization affects how you interpret data, for example, and helps you interpret how cells actually exist in the real world. And it kind of highlights the fact that a lot of biological processes are actually inherently random. Um, and so Braden will look into that with you guys. And what's really important I want to mention, actually, so this is going to be a shout out. So Professor Brian Ingalls is one of my mentors back at the University of Waterloo when I was doing iGEM there. So he's published this book called Mathematical Modeling in Systems Biology, an Introduction. It's a free textbook on kinetic modeling, on modeling metabolic networks, and it's really introductory. He walks you through kind of the intuition, the conceptual building, and the model building for a lot of this mathematical dynamical modeling. And so I highly recommend you look through this textbook. Braden also recommends it. He'll be using some of it in his lectures so that you can kind of enter the field of dry lab work in synthetic biology. So more specifically for protein engineering, when I say protein engineering, what we're trying to do is basically modify the protein in some way so that it has more useful features. For example, you might want it to have a higher enzymatic catalytic rate. You might want it to be more thermostable so that it operates at higher temperatures. These are some methods that you can use to do this. The first method is directed evolution. It's not necessarily a mathematical computational approach. But what direct evolution does allow you to do, which Marius will talk about in the wet lab module, is evolve a protein through rounds of mutations. And in each round of mutation, you select the mutation that encodes for an enzyme that has a behavior, has properties that are closer to what you want it to be. And so you kind of go through a landscape of different mutations that will eventually lead you, hopefully, to this, what we call absolute maximum, the best performing mutant. Now, the whole mutational process is a bit random because you're making these mutations randomly. What some people like to do is go through a more rational design approach. And so proteins, if you have their structure available, their PDB file available, you can actually look inside the protein and look at these different individual amino acids. And through our understanding of these proteins, through what's already been published, we can get a sense of which amino acids we might want to mutate to, and to which amino acid we want to mutate it to, so that it kind of gets a different property that we want it. And so QMMD, which is quantum mechanical molecular dynamics simulations, these are simulations that allow you to predict how the protein will behave and interact with other proteins with your substrate that you want it to turn into a different product, or even with metals as well. And so QMMD simulations are big in this in this protein engineering, and Tanya will talk a lot about this. And so Tanya will also talk about the machine learning methods that underpin some of the more cutting-edge protein engineering methods. And he'll kind of overview some of the different methods you can take in machine learning to do this process of protein engineering based on what information is already available in literature and in the databases. So let's shift gears a little bit from the mathematical computational stuff towards the more ethical and social considerations behind the Artemisinin project. So as I said before, the Artemisinin project is clearly a success. It produced an economically and commercially viable living biofactory that could produce literal tons of artemisinic acid when you do it semi-synthetically. It's a success, but one of the issues and kind of criticisms that have been kind of put into the online world, which I'll summarize for you folks, are the issues related to how complex the artemisinin market actually is. And the market isn't just comprised of a university doing research to produce this genetically engineered microorganism to produce artemisinic acid that Sanofi will produce, and then that somehow gets straight to the consumer, the patients that are affected by malaria. It's more complex than that. There are key players in this, including farmers that already produce the Artemisia annua plant for the pharmaceutical companies to make into ACTs. There are governing bodies that help form contracts and deals between farmers in these developed countries affected by the malaria with pharmaceutical companies to supply their Artemisia annua plant. There's more. And so what I'm going to do is walk you through how these two issues I'm going to talk about real quickly come into play for the Artemisinin project. So two things I want to quickly summarize are the stakeholder analysis and engagement. Whenever you're translating bench scale work to real world application, you need to look at how that real world application will affect the existing ecosystem, the existing market of stakeholders. And so stakeholders, as I said before, is a very key term here in practices. You want to identify people that have an important opinion that needs to be heard in this design process, in this translational process. And another key thing that kind of plays a role in this Artemisinin project is this idea of science communication. How well do these researchers communicate what their intent is, what their research is, to the general public, to the people who are affected by malaria, who are going to take these ACTs derived from artemisinic acid produced by their engineered microorganism. And how does public opinion play a factor in this? How is public opinion about these ACTs formed? Is it through fear? Is it through information? Is it through some kind of consent process to make sure that farmers are okay with losing the ability to make these crops because Kiesing in his lab and in Amherst is producing it semi-synthetically? These are important factors in bringing the product to life and translating it effectively into the market. Now, it's clear that what we're going to see is that the Artemisinin project took years to develop, and in these years that it developed, a lot of different key players, stakeholders, were also developing too. And that kind of impacted how semi-synthetic artemisinin was taken up by the market. So let's dive into that. To help us understand the Artemisinin project from an ethical and a social perspective, what we're going to use is an online debate between Jim Thomas from the ECT group in Canada and Rob Carlson from Bioeconomy Capital, who's a synthetic biologist. And through their online debate, we'll explore some of the ethics and trade-offs of semi-synthetic artemisinin, which I short form with SSA. So in this slide, we're going to go through some of the context behind semi-synthetic artemisinin. So this context is given by Mark Pepler, who published two newspaper-style articles in the journal Nature. So one of the first things was that to produce ACTs at the time when this whole Artemisinin project started, pharmaceutical companies had already made arrangements with farmers to source their plant to get the artemisinin, to extract it to produce the ACTs. And so these markets already existed in Asia, Africa, South America, the places that were affected by malaria. And big pharma like Gilead Pharma and Cipla in China and India, these are companies that have already established their market and established their supply chain. So when the project started, the Artemisinin project started in 2004, the goal was to drive down the price of ACTs so that it was less than $1, making it very cheap. And 8 years later, as the research has eventually neared its end, it turns out that the price of plant-derived artemisinin of these ACTs is already 92 cents. And this is despite inflation. And so what this is saying is that there was basically a big supply or a change in the supply and the change in the stability of getting the plant needed to produce the artemisinin. And so in 2015, it turns out that there's a big surge in the supply for this plant, and in fact, there's no SSA being produced by Sanofi. And so it makes you think about you embark on this long journey to produce this good, but you have to also remember that there's a market that already exists that can accept this good, and so you have to factor in the market entry and how that will happen and the economics behind that. And so speaking of economics, there's some weird economics at the beginning. It's kind of unclear. Sanofi was pricing their artemisinin at $350 per kilogram, when the plant-derived version was only $250 per kilogram, or even less. And so there's a trade-off here in terms of you buy this product from Sanofi, And sure, you have a more reliable supply of it, but the plant version is much cheaper. And so the market drive to have the semi-synthetic artemisinin is a bit questionable from this point of view. And lastly, as I said, this Artemisinin project has taken many years in the making to get to the final point where Sanofi would industrially scale it up. And so this is like basically a decade. In this decade period, other groups are also fighting malaria. And so the A2S2 initiative was created by UNITAID. And what they were doing was trying to basically do some logistical management of where the plant was basically being distributed, so that places that had shortages could actually get their supply, as opposed to just being a shortage and therefore having a rise in the prices. The A2S2 was kind of spreading out the supply of these plants. And additionally, there are other health organizations at the international level that were trying to instate better practices for diagnosing malaria. Because it turns out a lot of people were taking ACT drugs for fever-like symptoms, but it wasn't actually malaria. So improving these diagnostic procedures, particularly through the WHO's directives, it played a role in decreasing the demand for artemisinin and ACT drugs. And so as a result, when the SSA finally hit market, there were already factors in play that was decreasing demand for SSA in the first place. So this is the context that's actually going on beyond just the research that's going on at Amyris. And so now we're going to dive into a bit of the debate between Jim and Rob to kind of see what claims are made and what stances are made, and you can compare them yourselves and ask yourself what's right, what's wrong, and where can we improve future efforts to translate pharmaceutics into market. So in this first perspective, what we're going to look at is the hype behind SSA. Now remember that synthetic biology has been growing mostly through the past decade and a half. And so when semi-synthetic artemisinin was hitting big journals like Science and Nature, of course it's going to get a lot of media attention, especially because synthetic biology, there is more investors coming into play. And so of course they're listening to the news to see what exists out there that they can invest their money into. And so semi-synthetic artemisinin is a philanthropic thing, it's an economic thing as well that can produce a lot of money because you're working with pharma. So there's a lot of hype. And so behind this hype is Kiesling himself and Carlson. And I'm not saying that they generated the hype themselves, but from their point of view, they kind of support the push for semi-synthetic artemisinin. So here are some of their claims and stances, and you can interpret them how you think is appropriate. So one of the big claims that was made at the beginning was that by having semi-synthetic artemisinin and being able to produce this at scale, you can replace the entire supply chain for artemisinin, basically wipe out all plant production-based artemisinin and ACT drugs. That was the initial, and then they later kind of backed up and said, okay, we wouldn't do this all at once. We would do this a bit more slowly through our rollout adoption process that nations and pharmaceutical companies can kind of take on. And then much later on, they're talking about, okay, maybe we won't have SSA replace the entire plant supply chain, but just supplement some of the demand where in places that don't have the plant, we can supply it there. But remember again, A2S2 has already been doing this work to help properly redistribute those plants. So there's kind of competing interest here. Another thing is that SSA could be produced at 40 to 60 tons whenever needed. And so the idea was that this would saturate the market. And because it's saturating the market, it would kind of undercut the pricing of the plant-derived ACTs. That was the assumption. That was kind of the claim. And what we see is that that wasn't true. Just because the plant-derived ACTs, the price was already actually pretty low, and it actually became lower over time because of a decrease in the demand. One of the other claims and I guess stances from Kiesling and also Carlson is that by having Sanofi be able to produce SSA routinely in demand, you would actually be able to counter the black market for ACT. And that was because back then there were some knockoff ACT drugs that weren't full treatment being sold to people that weren't actually effective. And so by having Sanofi produce everything, all of the SSA, you would have theoretically much higher quality treatments that people could rely on better. And because it's cheap, they can access more easily. Again, this is a stance. Now, kind of tied and entwined into Kiesling and Carlson's perspective and what we'll talk about next, Jim Thomas's perspective, are farmers. And that's why I title part of this section C with farmers. Farmers grow all sorts of crops, and sometimes they will grow these cash crops like Artemisia annua for the pharmaceutical companies because they can make an extra few bucks. What Kiesling was saying is that, sure, we'll eliminate the plant growing market for supplying artemisinin, and so these farmers can just grow other things like potatoes, peanuts, rice, rather than artemisinin. And you can kind of see how this affects the livelihood of farmers who aren't able to grow some of these crops based on their geographic location. So how do you deal with that? And their argument was, sure, we might affect the livelihood of a few thousand farmers, but from a utilitarian perspective, having SSA being supplied to all sorts of countries in a controlled manner, in a predictable manner, could save theoretically millions of lives, or at least improve the livelihood of a lot of people. That was, and this is the Kiesling and Carlson's perspective that I wanted to show. So you can kind of see that this is just one side of the argument to not support our farmers in this case. And there are claims that SSA can kind of take over the market when what we see next, this isn't true. So let's look at the other side of the debate. And this is Jim Thomas's side, I would say. And what this side's going to do is kind of oppose some of the claims and stances made by Kiesling and Carlson. So these are the claims and stances by Jim Thomas. First of all, Carlson and Kiesling said that by flooding the market with ACT drugs derived from SSA, you could actually undercut the cost of plant-derived SSA and therefore sell it at a cheaper price. And so theoretically, that's good. You can, I guess, for the pharmaceutical company, you can kind of be the big player in this whole market. But the issue with fast introductions of drugs like ACT is that you actually increase the price volatility across international markets. And this is because the market for ACT is kind of heterogeneous. Different countries have different demands. They have different suppliers and distributors and producers of the ACT drugs. And by flooding the market, some places will get it at different times. And so you actually get a lot of volatility in places downstream that don't get it firsthand. The second point is that there is some claims about how making semi-synthetic artemisinin would drive down the price of ACTs, but the counter-argument is that artemisinin only makes up 40% of the cost of the ACT drug, according to Thomas. So any claim about it being cheaper than plant-derived treatments is kind of questionable. And another comment, so Carlson and Kiesling, they said that, or at least they were defending pharma companies who were saying that by producing semi-synthetic artemisinin, you could actually counter the black market for ACT. But of course, pharma companies are going to say this, according to Thomas. They're going to say this because they want to monopolize these markets where there's millions of patients that need ACT. Of course, you can make money off of that. Of course, you want monopoly. Of course, you want you to be the only supplier of high quality ACT drugs. And so there's a philanthropic drive in the artemisinin project, but there's also a financial aspect to it that might seem unethical, that might be questionable. So it's always important to balance the business demands, but also the philanthropic demands. And some other claims and general opinions by Thomas on synthetic biology. So the semi-synthetic artemisinin, by producing more of it and producing it in demand, when you want it, this is a response to malaria. Whereas there are other things that lead to more people getting malaria. And so Thomas kind of emphasizes the precautionary principle of really focusing on the source of the issue. And you could think of it in this case as improving the healthcare systems of these developing nations so they can better handle their malaria patients, improving diagnostics and treatment regimes. These are all things you can do to minimize the demand for malaria. drugs in the first place and the incidence rate. And the other point was made regarding the opinion that if you want to predict how people could use synthetic biology for unintended bad purposes, what you have to do is, as a good nation that is responsible, is for you to do all the research that in a controlled manner without malintent, but see how far you can take synthetic biology. See how badly it can be used in a controlled manner where it doesn't actually cause harm in order to prevent wrongdoers. And so Thomas was taking a cautious approach and saying that we shouldn't be doing this. We shouldn't just randomly and blindly push for innovation. There needs to be policy mechanisms to help safeguard what we call biosecurity, ensuring that these biological inventions don't cause harm to us and to the environment. And the last point that was made is the fact that when synthetic chemistry emerged earlier in the 1900s, it turns out that poorer countries were actually became poorer because they weren't able to supply their natural versions of these chemicals that can now be produced synthetically in factories. And so who's to say that this won't happen to synthetic biology? So to kind of bring everything back together and summarize my point, you can see that the Artemisinin project kind of has different takes to it depending on who you are. So I encourage you to look back at this slide and the previous slide and compare these two, talk about it, look at the actual articles that Jim Thomas and Rob Carlson wrote, and get a sense of who's right, who's wrong, and where we can improve for future versions and iterations of this kind of work. The stakeholders in this situation, you can kind of see all of them here. These are types of stakeholders. So the stakeholders in the situation for the Artemisinin project include people who funded it, who are doing the research, and who are going to sell the actual drug. And it also includes those who actually live with the malaria, their families, the neighboring communities. It includes technology watchdogs and whistleblowers who may have some particular insight about a certain market. There are also governing bodies internationally or nationally, such as the WHO, A2S2 in this case, and other health organizations. It's important to consult with them because you want to get their insight into the patients and what are the issues that they're going through. Maybe it isn't just access to ACT drugs. Maybe it's improving diagnostics, as I've been reiterating, which was one of the key things that drove down the demand. It was better diagnostic power. And lastly, the media is incredibly important here. The scientific journals, the press. In my search to find material to teach this particular SYNB0 lecture, what I was looking for was some kind of communication from Amaris, from Kiesling himself, that was publicly available, that I could look at and be like, okay, these are the intentions. And so obviously the scientific papers that are being published by Amaris, by the Kiesling lab, generally talk about the goods behind this kind of technology in their introductory section, but this isn't accessible to regular people, to people who might be affected by malaria. What's really important is that when you're trying to bring these technologies to life, to translate it to applications that affect people's livelihood, it's really important to get perspectives from people that you're actually affecting and downstream stakeholders. And this is because it might influence your design process. In this case, I don't know if it affected their design process or not, if it could have. But in some cases, you can imagine where you don't want a microbe that was genetically engineered to be escaping into the environment. Well, in that kind of situation, you want to put some kind of genetic mechanism to ensure the bacteria doesn't escape into the environment so that it dies without having something that is only existing in the laboratory. If we knew there are regulations that limit the spread of GMOs in the environment, which there obviously are, that would impact the way we do our experimental work, our modeling work. So again, from this big list of stakeholders, who are the most important people who actually play a role in implementing semi-synthetic artemisinin? Well, it's obviously going to be the first 4 people who are funding it, doing the research, and distributing it. But really, I want you as an emerging synthetic biologist is to consider who is going to be affected by this downstream. And typically people who have better knowledge of that aren't the people who are funding and doing the research. It's people who look after the people who are dealing with these problems. So in this case for malaria, it's going to be these watchdogs, it's going to be these health organizations. It's very good to consult the people downstream of your application and your work. So let's bring everything back together. So I'm going to share with you again this map of where malaria affects people in the world. And you can see malaria has far-reaching consequences on a lot of people's livelihoods. And there are a lot of issues besides malaria that also have far-reaching international implications. And synthetic biology, being such an interdisciplinary field, has a huge potential to address a lot of these issues. And I'm not saying synthetic biology will solve everything. Synthetic biology will produce some technologies to make major impacts on these issues in conjunction with working with other stakeholders. And so right now I want to bring back the definition of synthetic biology that I offered by combining everything in section B. So take a pause in this video and look through this and read the yellow highlighted parts with detail. So what I'm doing by highlighting these parts is I'm trying to emphasize what the Artemisinin project did really well with. And what I'm going to do now is highlight some of the purple parts, the parts that the Artemisinin project could have benefited more from. So in particular, it could have benefited from more computational simulations, more computational methods to do the metabolic engineering, that pathway engineering. And it could have used more social sciences and ethics in developing some of its downstream applications and entry to market in that plan. And a truly synthetic biology project is something that combines the experimental with the modeling to improve that experimental workflow and brings it all together and translates it to society using better social sciences and ethics by understanding how this technology that we're creating, these living robots, these living biofactories, how they will affect the livelihoods of people, of society, and of the environment especially. So Artemisinin isn't the only thing that can be produced through synthetic biology. As you might be very aware of, synthetic biology can be applied through all sorts of things that we also see in the iGEM competition. I've judged the competition for 3 years now. I've been on the team for 2 years. The 5 years of iGEM, I've seen all sorts of projects, and I've seen the variety of issues that can be addressed through it if we just carried through some of these projects to entrepreneurial levels, to levels where there's more commercial viability being explored. And so iGEM is an amazing place. There's so many people that you meet through iGEM, and you really learn a lot about what you can do with synthetic biology, what good you can bring to the world. And so I want to introduce the 3 streams that we want to talk more about through our 3 modules. And so one of the first modules is human practices. So in human practice, we explore the social and the ethical dimensions of synthetic biology. In particular, we look at how to analyze problems, stakeholder engagement and outreach, as we've addressed through the Artemisinin project and its importance, science and mathematics communications, risk assessment and the determination of risk, and policy development. So Estee and Umar are going to explore these 5 topics with you through their instructional material, so I point you to their direction. So for the wet lab stream, for this module, we're of SYNB0. We're going to divide up some of the topics into the DNA focused stuff and the protein focused stuff. So the DNA focused stuff I'm going to primarily teach, and so I'm going to talk a lot about some molecular cloning basics, and then I'll do some walkthroughs for bioinformatics tools you might use on a regular basis, being an experimentalist in the lab. And we'll also give an overview and introduction to benchling, which is what we recommend for managing some of your DNA sequences and protein sequences. And it has a variety of tools to help you with sequencing with CRISPR design. So we'll kind of give you an intro to how to use benchling. And Marios will be teaching some of the protein-based stuff. So CRISPR gene editing is one of the first topics. It's focused on editing DNA, but it's also a protein that's doing it. So Marius is going to talk a bit about the mechanics behind how that works, so you have a better understanding of how CRISPR works at the molecular level. He'll also talk about directed evolution, and talk about enzymes, how you can purify them, see them better through structural determination, and understand their behavior through enzymatic binding assays. For our third module, we have the dry lab. So Brain is going to be going over all sorts of topics, introductory topics, for fundamentals of modeling biology. Kavya will go over some bioreactor systems, an overview of them, a few case studies, so that you can better understand the economics behind how these bioreactors work. And lastly, Tanyu will go over the computational protein design from the rational design perspective. So he'll go over molecular dynamic simulations and some machine learning methods to help design these proteins. Okay. So again, the three modules are human practices taught by Este and Umar. We also have wet lab taught by myself and Marios. And we have dry lab taught by Braden, Kavya, and Tanyu. So please look forward to those modules that will be uploaded very soon and learn a lot from them. Take time to go through them. They might be a little bit long, but take breaks as you go through these lectures. If you ever have any questions along the way, please feel free to ask us by contacting us through our email. It's in the description below, or just comment on the video themselves and I'll alert our instructors of your questions and they'll respond accordingly. And lastly, as you go through this lecture material through SYNB0, remember that we're using this definition of synthetic biology. And we kind of hope that you agree with this, or at least agree with parts of this definition and use it in your own practice, remembering that synthetic biology is truly interdisciplinary. It's not just experimental, it's not just modeling, it's very much also social sciences and ethics playing a really big role in helping translate a lot of this technology. So again, on behalf of my CSBERG instructor team, we thank you for listening to this introductory, very long video, and we hope you enjoyed it, and we'll see you around our future lectures for SYNB0. Thanks again. I'll see you guys around. Stay fresh and stay safe. Cheers.